I'm very pleased that Adam Hartman could come in person. We're having a real seminar today with real food and real people, so glad to see you all. Adam is well known in the circles of studying motor systems as one of sort of the avant-garde of the new optogenetic wave of mouse studies that is taking the world by storm, and he's sort of one of the whirlwinds of that storm. He got prepared for this job through a bachelor's in biology. Since I'm a biology department, that always sounds great. In 1999, he got a PhD at UNC with Ed Pearl. He was already interested in the spinal cord and movement. A postdoc with Thomas Jessel, who we all, I think, know from the Jessel-Schwartz-Kandel textbook. That was at Columbia. And then he was a group leader at Janelia for 10 years until last year. And Janelia had this tendency to turn over and to send their people to new and better frontiers. So he went back to UNC, to his, I think, very PhD department, who I'm sure were super happy to fish him back. And now he's in his own lab there as associate professor studying how mice process movements. And without further ado, thank you so much for coming, and we're enjoying your seminar.
I want to thank Dieter for inviting me. I'm super excited to be here. I've been following a lot of the work that's been coming out of Georgia Tech and Emory for many years now, so I'm very excited about all the meetings today with the students and the faculty. My lab, as Dieter mentioned, is interested in motor control, and I think, at least my primary interest in here sort of stems from the fact that I think that motor control is one of the oldest and one of the most essential functions of the nervous system. It's really critical for all aspects of our survival. It's how we find our food. It's how we find our mates. It's how we avoid our predators. And because it's so old and so important, I think it might provide real insights to lots of other functions because perhaps they're built on the architecture of the motor systems. Now, it's true that we don't actually need a nervous system to actually produce movements. Of course, we know that single-cell organisms and very simple animals with no discernible nervous system can, of course, move through their environment. I like this example of this trichoplax here. It's a very simple animal, has nothing that resembles a nervous system, yet it can sense things about the world, move to those things, and then interact with them, at least by ingesting them. And so you can, of course, make these movements without a nervous system. So why do we have a nervous system then? Well, I would argue the reason why we have a nervous system is to make movements way better, to really upgrade our movements. It allows us to sense and respond to things from much further away, make complicated decisions about those movements, move faster with more control, more coordination, and to even use large effectors such as limbs and even tools for some species. And so one of the primary questions in my lab is, how does the nervous system afford us these upgrades? And the particular matter that we're interested in is for limb control. How does the nervous system take control of these limbs and use them for their behavioral needs? And of course, for the nervous system to do this, what it must have to do is understand the properties of these limbs, the ones that it has been born with. And then given that understanding with some goals in mind, it eventually has to do kind of a simple thing, which is to generate motor neuron output and deliver it to the muscles, give it a pattern of activity that will then hopefully move the limb in a certain way to achieve some sort of behavioral control. Okay, so it's pretty simple on that side. And so you can think about the rest of the nervous system, everything that sits upstream of these motor neurons, as kind of a pattern generator. All they need to do is generate the pattern of activity in the motor neurons. And actually, thinking about the nervous system as a pattern generator is a really old idea in the history of neuroscience. It goes all the way back to sort of the forefathers of the field, including Charles Sherrington, who was really interested in pattern generation for rhythmic movements, such as locomotion. And actually, originally, Sherrington thought that those pattern generators were actually peripherally dependent, that actually it was the sensory information coming back into the nervous system from the periphery that was actually driving the next steps of the motor output, that it had the information. Now, that was convincingly demonstrated to be incorrect by Sherrington's own student, Thomas Graham Brown, who took this question on, and he actually showed that the pattern generators for locomotion were actually central in origin. Actually, the spinal cord could generate the basic patterns for locomotion all on its own. You could deprive the spinal cord of its sensory inputs, of its descending inputs, and as long as you gave it a kick, it would generate some of the basic patterns of activity underlying the rhythms of locomotion. So my lab is actually also interested in these ideas of pattern generation in the nervous system, but not for these rhythmic movements, but actually for skilled movements. This is how we learn to interact with our world adaptively and flexibly. It is the movements that we get better with over practice. And of course, it's also the movements we spend a lot of money just to watch other people do. And it's because of this practical importance and because of their cultural importance that neuroscience has, of course, been studying this really for over 100 years. And what we've been doing as a field is sort of going through, so there's a little bit of a delay here. We've been going through all these different regions of the nervous system and then asking how each region might contribute to either the learning or the production of skilled movements. Now today, what I'm going to try to do is a little bit of a twist on this, where we're going to start to ask questions about how some of these areas actually interact in order to produce these skilled movements. And we're going to focus on 3 areas, motor cortex, thalamus, and cerebellum. And we're going to start off with motor cortex because it is thought to be one of the real master commanders for skilled movements. There's lots of reasons for thinking this. One of the reasons is that damage or disease to this part of the nervous system leads to really severe consequences, almost specifically in skilled movements. If you have stroke in this area, skilled movements are greatly deteriorated. This is, of course, the area that if you do stimulations of particular kinds, you can get very discrete movements, which are thought to be potential building blocks for skilled movements. And this, of course, is the region that has neurons both at the single neuron level and at the population level that have really rich information about skilled movements. And I think my favorite version of this right now comes from the work of Krishna Shenoy's lab. Krishna's lab has been looking at motor cortex and the population activity within motor cortex, and then examining what the behavior of that population activity is. So each one of these traces here represents the population activity within motor cortex over different movements, where every different trace is a different movement. And this is a state space plot. We don't need to understand this plot yet. We'll come back to it. But all I want you to really recognize here is for all of these different movements, motor cortex seems to be doing something a little bit different, and there's rich information in these dynamics. So, one could argue that actually it seems like we understand a lot about how motor cortex is involved in skilled movements, and I actually agree with that. I think we do understand a lot. But there are some really basic questions that still remain open. And there's going to be one that we're going to tackle first here, which is I'm sorry, is it a little bit laggy? Is it like jumping, the videos? I can't quite tell. Okay. The basic question is, where do these dynamics actually come from? Okay? Like who's generating them? And there's sort of 2 ideas in the field, sort of sitting at the extremes of the possibility. One is, much like the spinal cord, maybe motor cortex is generating those patterns all on its own. It's like sort of an autonomous dynamical system. It's capable of generating these patterns of activity. As long as it gets a proper trigger signal, it can generate those. And it's learned how to generate those, importantly, unlike the spinal cord, over the training process. And I would argue this is probably one of the leading ideas in the field, and there's good reason for that, and a lot of practical good reason for that. But there is another possibility, and it's probably an older possibility, which is that motor cortex is really critical for producing these patterns of activity, but actually it can't do it on its own. It's constantly relying on inputs from other areas, putting them together, building them up, this pattern of activity, but on its own it can't generate those patterns. And this is more of a distributed dynamical system. And I think it's really important to actually distinguish between these 2 hypotheses. Not only does it tell us about the true nature of the dynamical system underlying skilled movements, but it also tells us how to study this going off in the future. For example, if you're in this local autonomous mode, then perhaps you really should just understand the fine features of motor cortex and how it changes over the training process. But if you're in this more distributed dynamical system, then you're sort of forced to try to tackle the interactions of all of these other areas of input and how they all combine in motor cortex. And I would argue those are fundamentally different jobs to do. So how have we got into this place where we haven't sort of answered this very basic question, I think, about this system? And I think one of the reasons is that most of the pioneering work in this field was actually done in primates. And that has been excellent for electrophysiology. It's been excellent for behavior. But where it is a bit limited is our ability to do causal perturbations, where we affect some part of the nervous system purposely and then monitor the consequences both neurally and behaviorally. And I would argue that it's those classes of experiments that help distinguish between these two possibilities. So when I started my lab now 12 years ago, I decided I wanted to take on this problem. And I thought one way to do it was try to port these questions to another model species where we could actually take on these experiments. And the model species that we decided to work on was the mouse. There was a couple of different reasons for this. First of all, the mouse makes excellent use of all these new tools for doing causal perturbations, tools like optogenetics and pharmacogenetics. They were in part developed in the mouse, and they work quite well there. But we also picked the mouse because they can do really nice versions of skilled movements. And perhaps the champion of these movements, at least for the neuroscience community, was this movement. It's a reach to grab task, really developed by Ian Wishaw in Canada. The iteration that they had was freely moving animal sits in this box. There's a slit at one end of the box. And when the animal wants to eat a food pellet, he throws his hand through the slit, grabs the food pellet, and places it in their mouth. And when you look at the fine features of this movement, as I'm showing you here on the right, they seem to do it through this stepwise execution of lifting and opening their hand and then grabbing. And they'll do this double supination and go to their mouth. And what I really like about it is actually it looks a lot like actually how a human would grab an object off of a table. And so we love that sort of analogy to our own behavior. Now, when we started to think about this behavior, though, many years ago, we wanted to change it in one fundamental fashion, and that we wanted these animals to perform this behavior while being head fixed. Head fixation offers a slew of experimental advantages. It allows us to isolate the movements as much as possible to a single effector. It allows us to impose a cued trial structure on the animal, which was really critical for the work in the primate. And perhaps most importantly for my lab, it really facilitated these hopes to do recordings and manipulations from within the animal's brain. So we tried to develop this task over the last 10 years, It's kind of a simple task. A head-fixed animal sits in this rig, comfortable, we at least think. His hands are on this perch. There's a table out in front of the animal, at least in the first iterations of this. And after a tone and a prescribed delay, this table will rotate, deliver a pellet of food within the animal's reach, and then hopefully they'll reach out and grab the food pellet. Okay? Now, there's one thing I need to tell you about this behavior, which is kind of critical, which is basically done in the dark or blinding light to the animal. So the way that we think that they have to solve this behavior is they have to memorize the position of the food pellet, store that memory of that whole motor plan in their brain somewhere, and then call it up when we ask them to do the behavior. Okay? So actually, animals can do this, mice can do this quite well after training. Training can take anywhere from a couple days to a couple weeks, depending on the trainer and the animal. But they can do it really nicely. They understand the tones and the prescribed delays of the task, and then they can smoothly and coordinately execute through all the steps of this behavior. And these animals can really achieve remarkable success at this behavior. So, for example, if you look at trial after trial, which I'm showing you here, these are just consecutive trials. They can reach success rates basically on 90% of the trials. And this is the first reach attempt. This is about double the success rate you see in the freely moving version of the task. So these are just absolute all-stars at the behavior. So we really like the skillful performance that the animals are achieving. We really like the analogy to our own behavior. But there is one sort of annoying thing about this behavior, which is it's best captured through video. There's no sort of analog readout of this. And back in the early 2010s, when we started to think about this, we thought that the way of analyzing these videos, which was really through manual annotations, were going to be too time-consuming and laborious, really, to really last that long in the lab. And this was around the time when computer vision and machine learning was really taking off, and we thought that we had a pretty good task for computer vision. We had this fixed registration to the cameras, a nice stereotype trajectory. And so we teamed up, actually, with a really brilliant computer vision specialist at Gedalia, Kristen Branson, and we presented this problem to her, and she thought it was completely solvable, and she was right. And so over the last 10 years, we really created a suite of tools to help us analyze all the features of these videos that we were interested in. I'll just mention these briefly because these have become sort of old hat now in the community. We have one toolbox that basically can convert these videos into ethograms or graphical depictions of the movements, where we basically make up classifiers. I don't know why these little things are coming up. Graphical depictions of all the different steps of the behavior, as you can see down here. You basically train up a classifier for these, and we also have ways of actually monitoring in 3 dimensions positions of any unmarked part of the body. And these things have been great. They not only help how we analyze our videos, but they also fundamentally change how we do our experiments, because you can actually run these in closed loop with minimal latency, and that allows you to detect features of the behavior and then go back and perturb the animal any which way you want. And that will be critical, as you'll see in a few slides. So we were feeling pretty good about ourselves, had this nice behavior and this nice analysis methods. And so we thought we could start really asking some fundamental questions about how cortex was actually pulling this behavior off. But we were presented with maybe not an unexpected problem, but an annoying one, which is that the field was sort of losing faith that rodent motor cortex was really doing the same thing for skilled movements as was the primate motor cortex. And a lot of this doubt really came from chronic lesion experiments, such as this one. This is a very large lesion of rodent cortex, actually extending even beyond motor cortex. And while there is a persistent deficit in these lesions in the skilled reaching test, the freely moving one, the effects are much milder than you would have imagined. They kind of fumble the food pellet on more trials, but they're still able to do the basic steps of the behavior. Now, I think that these chronic lesion experiments are very, very interesting. I think they can tell us a lot about the brain. I think they can particularly tell us about the compensation capabilities of the brain. What can the rest of the brain do in the face of the loss, in this case, of motor cortex? But what I don't think they necessarily tell us about is what does motor cortex do normally in an uncompensated brain? And I think if you want to get at that question, well, then one way to do this is to try to find a way to minimize compensation while taking these brain regions offline. And so you might want rapid, reversible, unpredictable forms of perturbation. And of course, optogenetics is ideally suited for this. So back in the day, for the first time ever, we wanted to use optogenetics to turn motor cortex off during these reaching assays and see what happened. We've done this in many different ways now, but the original way we turned off motor cortex was actually by leveraging a mouse line made by Guopin Fang, who made this mouse line that expresses channeled opsin in the inhibitory neurons of the cortex. Expression of that opsin and exposure to blue light activates the inhibitory neurons, and they dump GABA all over motor cortex and shut it down locally. And the nice thing about this is you can do this on random intermittent trials, so the animals will compensate for it less, and you can do it at whatever temporal epoch you want. So at first what we're going to do is just sort of do it over the duration of an entire trial, but on random intermittent trials. And because we wanted to map out sort of the full cortical space here, we're going to do this over many cortical regions. And actually, most cortical regions, silencing it gave us absolutely no effect. And I'll just show you one example of that. This is ipsilateral sensory motor cortex. The laser's on here. We've delivered the food pellet, and the animal's able to perform the behavior really unaltered in any way that we could detect. Okay, and I think this serves as an important control that if you ever do see an effect with blue light in these channeled opsin expressing animals, it's not from the light or some non-specific effect of shutting off a very large swath of the brain. Okay, so this is ipsilateral sensory motor cortex. You wouldn't imagine maybe it would have a huge effect, but you might imagine that on the contralateral side to the arm, you'd have many areas that were involved. Actually, we found very little effects except for in motor cortex. And here the effect was absolutely dramatic, and that if you turned off motor cortex on the contralateral side, the animal simply could not engage in the behavior whatsoever. So for as long as we hold this laser on, you're going to see the animal's simply not going to move. And our animals never miss an opportunity to reach for a food pellet. They are fairly hungry. Now, we were a little bit worried at the time that this had actually nothing to do with the movements of the arm, and just that the animal didn't understand this kind of weird trial structure we had set up for the animals. So we decided to quickly probe that by putting these same animals in a context where the trial structure is exactly the same, but they could actually do a simple ballistic movement of their tongue in order to consume the food pellet, just a simple lick task. And you can see here that turning off in these same animals, this part of motor cortex has no effect on their ability to solve the task with a simple ballistic movement, as you can see here. Now, I want you to notice something about this video, though, and I can replay it, because I think it's really interesting. I think this is like a general statement I want to make about looking at all the videos, because there's rich information here. While the animals are able to sort of well time the task here, they are also able to actually move this normally affected hand. This is the hand that they would normally reach out for. It's the animal that the hand that they could normally not initiate the movement at all. But in this context, they are able to initiate the hand movement. It's actually not that different. They're going to lift, they're going to supinate, they're going to go to their mouth, they're going to help to push the food pellet into their mouth. But they are able to initiate that movement with the motor cortex off. And this and many other experiments suggested to us that there was something very specific about this learned skilled behavior, and maybe the learned skilled interaction with the world, which is the major difference between this task and the other one for the hand, that it really makes you sort of dependent on the cortex. All right, so this really was suggesting that cortex was absolutely required for the initiation, specifically in these learned skilled behaviors. But is that all it was doing? Was it just important for initiation, or did you actually require motor cortex past the initiation for the continuous execution of these movements? And actually, this was a very long-standing question in the field of motor cortex and motor control. Yep. Yep.
Just a question. This looks pretty bimanual to me. Did you find that the kind of movements that survive, motor cortex being silenced were more likely to be bimanual than?
I don't know if I don't have enough data to a real Olympics of arm movements and really test it out. I will say, movements that definitely survive are movements like scratching, locomotion, this one. Now, when he's initiating that movement from the perch up, that his other hands are actually doing something very different. So it's not a coordinated, sort of synchronous movements of the two arms, and it's still affected. And then many people have tried to silence it through simple joystick tasks, and you don't stop the movement. But those are also frequently bimanual in actuality. They present it like a single, but they're actually, animals are usually doing that. Okay. So bimanual movements, there might be interesting cases for bimanual movements when they're doing interactions. We haven't probed it good enough yet. It's an interesting question. There's been lots of hope of on a transfer task that might be really cortically dependent, but we don't know yet. Yep. Okay, so the question is do you require cortex for the continuous execution? The reason why this hasn't been probed even in the monkey is you need to do this kind of special experiment where you let the animal start the behavior unperturbed and then detect the movement and then perturb somewhere midway through the behavior. That's pretty hard to do, but with the rapid sort of on of optogenetics and the closed-loop nature of our experiments, we're able to try to do this experiment for the first time. And this was, it sort of gave us superhero-level status over these animals because we could essentially freeze or pause the animal almost anywhere along their path. And I'll show you two examples of this. The first is we're going to turn off motor cortex during this ballistic form of the movement where they are basically lifting off the perch and going out to the food pellet. It's thought to be too quick for sensory information to guide it, so it's thought to be largely pre-planned. And what we're going to see is as soon as we turn off motor cortex here, the animal's arm is going to stop in midair, so right there. And you can see that trial over trial over trial. They just cannot complete the task, and we've never seen an animal stop midway through a reach before. All right, so motor cortex seems to be required for this sort of ballistic component, but what about when you get sensory information? Can the sort of sensory feedback sort of take you home through subcortical systems? For that, all we're going to do is hold back the perturbation until after they're getting rich sensory information. In this case, actually, we're going to let them grab the food pellet and have the food pellet in their hands. And what you're going to see is, again, they cannot complete the task. So the animal reaches out, grabs the food pellet, then we turn off cortex, and his hand gets stuck here and holds in this position. Now, again, I want you to notice something about this video. This animal is not disengaged from the behavior. He's actually trying to pull up his affected hand with his unaffected arm. And he's trying to complete that task, but he's still, the hand is stuck. So obviously motor cortex seems to be critically important for moving this hand, but I think it's important to think about is that the rest of the brain is still ticking away and trying to find a solution to this problem. And I think we should just remember that in all of these optogenetic experiments. And I will just mention that, again, this is also very selective, the interruption of an ongoing movement, selective to this skilled movement. If you do it during grooming, for example, the animal is unaltered. So this was sort of building up a nice necessity story for us for cortex. It seemed to be necessary for the initiation and the continuous execution of the movement. But in these same experiments, we also started to gain some evidence that perhaps manipulation of cortical activity was sufficient to at least evoke the movement. And the evidence for this really came from watching what happened right after we stopped inactivating the cortex. Sorry for that double negative. But what we saw is when we flick from laser on to laser off, and laser's been on for 2 seconds, I just have clipped it. Every time we do that, the animal immediately reaches out for the food pellet. Okay, and so we thought maybe he remembered the food pellet, or maybe he resensed it during this inactivation period, but that didn't really explain all of the cases. And there was something kind of weird about the timing of these responses. If you looked at the latency from when the laser went off to the production of that movement, the latency was shorter and tighter in its jitter than was the normal behavior in relationship to the tone, which is what the animal learned. Like that's the thing that they had learned. And this started to suggest to us that maybe the actual perturbation of cortex here and its release from that perturbation was actually causing the movement to happen. So we wanted to test that. We thought one way to test this was to take an animal, put them in a completely unmotivated state for reaching. No food pellet, no tones, nothing. All we're going to do is perturb their cortex and then release that perturbation and see if that causes reaching. And lo and behold, this perturbation, and actually now many other perturbations, was effective at causing the animal to reach out where the food pellet should have been. And actually, if you slip a food pellet into position, they'll actually complete the entire task. Okay, so perturbation of motor cortex and released, like that, somehow affects neural activity. And instead of going back to rest, it actually goes back to the movement. So perturbation of motor cortex is at least sufficient to evoke this entire motor program. So I thought at this,
Yeah, the question, does that only work in well-trained mice? After how many days of training does this effect occur?
Yep. So we've tried to map this. It's a little bit difficult. The question is, does this only work in well-trained animals? It only works in well-trained animals. It actually seems to only work once the animals know not only how to reach well, but also when they have minimal latency and variance on their normal initiation time. Like when it becomes like so hardwired in there, they're basically going at max reaction, or min reaction time, sorry. That's when we start to see this. But then their reaction time to the laser is actually about 100 milliseconds faster than that. So it's, some animals will go in 50 milliseconds after the end of the laser. Any other questions? Yep.
Context dependent is it? If you put them in a different room such that they don't know that they might be presented with a treat, would they still be able to evoke this?
Yeah, so we have the question is how context dependent is in a specific experiment if we put them in a different room. So in their little cradle, because we don't have any other way to do it, but in their cradle, we have taken them and put them in the 2-photon, which is a very different arena, taking the table out. So now they've never seen this arena and there's no table to even orientate them. They will reach in midair. Now, we've never done it freely moving or things like that, but there are other weird vagaries of this particular behavior. So, for example, if you tried to do this within 20 seconds of the last successful trial, where success is measured by just the pellet touching the tongue, even if they don't consume it, then you cannot evoke it. If you take them off food depth for 1 day, they will still reach to the cue on every trial, almost every trial, but they will not do it to the laser. So there is some weird dependencies here that we don't fully understand. Yeah. Okay. So anyway, the basic, this is just to get to this point where now I think, at least for us, we feel confident that motor cortex in the rodent seems to be pretty important for skilled movements. And, that might seem like a small win, but it was a big win at the time. And that gave us some confidence that we could start to ask these questions now about how motor cortex was actually doing this, how it was involved in these behaviors, and maybe that would have some relevance for the work in the primate. So this brings us back to this question about where are these beautiful patterns of activity coming from in motor cortex? Well, before we get to that question, we do need to confirm that there are beautiful patterns that are highly related to the movement in motor cortex of a rodent. So we're going to leverage the head-fixed nature of our experiments to establish electrophysiological recordings while the animals are doing the behavior. We wanted numerous simultaneous recordings from many units so we could get an assessment of the population activity. So we're going to put down these silicone electrodes with high-density electrode arrays deep into the cortex. We're going to target layer 5, which are the output layers of the cortex. And you can see down here, while the animals are performing this movement, we have a subsample of the electrodes here, and I've colored in units that we think are individual units. And you can see nice recordings here and some relationship of the firing properties of these neurons to the actual behavior. But let's dig in a little bit more. There's just a few trials. So here are 4 representative units of things that we see in motor cortex. Histograms of their firing rates are on the top here, then raster plots are on the bottom. In these raster plots, every one of these rows is a trial, and every little tick is a spike. Okay, so you can see here a real sort of zoo of neurons, even in these 4 here. You have some neurons who really like to have an increase in firing rates in relationship to the behavior. This is all lined up to the production of the movement where the animal's first lifting their hand off. Some neurons, interestingly, turn off right before the behavior or right during the behavior, and then other neurons have more complicated relationships. So real diversity across at least this very small population, but I want to make an important note here that within a neurons, they are remarkably consistent across trials. They really are doing very similar things across all these trials. But this is 4 units. Let's show you a bunch more units. This is 600 units, which used to be a lot. Now it's a small fraction of the population that we have. This is a heat map of the average activity profile of these 600 units. Every row here is a neuron lined up to the production of the movement. And then what I've done is I separated these neurons out into 3 major categories: those that increase their firing rates, those that decrease, and then still other ones that, at least by this statistical test, are not modulated. So the first thing to notice here is, even without hunting for neurons, like we just put the electrodes in and we get what we get, 70% of the neurons at the ends of our electrodes really care about this movement. This area fundamentally cares about the behavior. A little bit over half increase their firing rates, and a little bit under half decrease their firing rates in a kind of interesting way. But let's see if there's a pattern of activity here, okay? Let's concentrate on these neurons that increase their firing rates. And what I've done is I've sorted these rows, which are the neurons, by the peak of their firing rates. And what you'll see is this really nice sort of tuning of neurons to different aspects of the behavior. Some have their peak firing rates before the behavior starts, and then others sort of tile over this whole behavioral expanse. And so perhaps these are involved in initiation, and these are involved in this sort of continuous execution, and are possibly why we can stop the movement midway through. Okay? So this is at the single neuron level, but of course, we really want to examine this also at the population level. This is really inspired by the work from Krishna Shenoy's lab. Population activity is a little bit difficult to visualize because it sits in high dimensional space. It's related to the number of neurons you're recording from, and now we're recording from 300-400 neurons at a time. So what most people do for visualization purposes is to do a dimensionality reduction. You can think about that of like finding like sort of super neurons, and then you basically plot the lowest dimensions of that dimensionality reduction against one another. Then you can sort of map the trajectory of these super neurons through neural state space. So that's what I'm going to show you here. On the left is the 3 dimensions that explain most of the variance in mouse motor cortex. Every one of these little dots here is going to be a different trial for this particular session, and how the neural trajectory unfolds through the state space. And on the right, for those same trials, what's happening to the arm in 3 dimensions in the real world. And then these little gray things are just the projections on the backfields of the arena here. And so what you're going to see here when I play this movie, if it plays, is you really get this real beautiful pattern of activity in this state space plot on consistent over trial, and it's heavily related to the information on the right. Matter of fact, I can decode what's going to happen on the hand from what's happening on the neural trajectories. And this dynamics is really important. If it happens, and I don't have time to show you all this data, if this dynamic happens, it's going to reach. So it's sort of, if this thing is going on, the animal is reaching. It basically will look like this no matter how we ask the animal to reach. So, for example, these blue traces are actually these laser-evoked reaches. So the animal has never even heard of a sound or initiated it in that same way, yet the trajectory is the same. And if the trajectory fails to get up this hill and around this corner, the animal's not going to reach. Okay, so these dynamics seem really important. And this then finally allows us to get back to this main question about exactly where are these dynamics coming from? Are they autonomous and coming from motor cortex, or are they from this more distributed system? Now, the critical difference between these 2 hypotheses is really the role of the inputs. In this local autonomous mode, all the inputs are doing is triggering motor cortex. And motor cortex goes and does its thing. And in this distributed system, the inputs are constantly important because they're helping build up this pattern of activity. So what we wanted to do is manipulate these inputs and ask when and how they were important. So there's lots of inputs to motor cortex, but the major input, of course, comes from motor thalamus. This is just the parts of thalamus that project to motor cortex. So we decided to manipulate those first. We've done this many different ways, but the original way we did it was by actually co-opting their inhibitory input, which comes from a nucleus called the thalamic reticular nucleus. We put channelrhodopsin into that nucleus. We put fiber over motor thalamus, shine blue light through it, that activates those inhibitory terminals, dumps GABA all over motor thalamus, and then shuts it down. That's been shown by us and by others that works quite nicely. And so then the first thing we did was just shut off motor thalamus on random intermittent trials throughout the whole trial, and they really have a hard time initiating the reach altogether. You can see the lift probability here is significantly decreased. And this really suggests that motor thalamus is actually really necessary for this behavior. But it actually does not distinguish between these two hypotheses because motor thalamus could be the trigger in either one of these cases. So what you really want to do now is hold back the thalamic perturbation until after initiation and then see if you can stop it midway through the reach. That's what I'm going to show you here. This is the hand position, at least along one of the dimensions on a control trial here in magenta. And when we do the thalamic inactivation, that's going to be this white line that turns into green line when we do the thalamic inactivation. You can see as soon as we turn that thalamic inactivation on, the animal's arm stops and either holds in a certain position or regresses back to the starting position. Okay? So this is suggesting that thalamus is required here throughout the continuous execution of this movement, but why? Why is it required? You can imagine two different possibilities. One possibility, which is kind of boring, which is that actually thalamus is not part of the pattern generator, but it does provide a tonic permissive signal to motor cortex. And without this tonic permissive signal, motor cortex slips below its threshold. And it really is the pattern generator, but because it's below threshold, it can't exhibit it, and motor cortex is just silent. Okay? Now that's in opposition to a more interesting possibility, which is that motor thalamus is critically part of this whole dynamical system, part of the pattern generator. It's got rich information about the pattern in motor cortex because it's building it. And without this thalamic information, it's not that cortex goes silent, but actually you just generate a bad pattern of activity. All right, so let's see which one of these is true. Well, what you have to do is actually record from motor cortex while doing these thalamic perturbations and ask what regime we're in. That's what we're going to do here. This is now trialed average PCA, sort of like those dimensionality reduction plots I showed you before for the trials. We just sort of averaged over all the trials. And this is what happens in the control situation. The neural, I just briefly mentioned that we're doing this from this period of time where we actually evoke it from the laser. We did that so we can always get the neurons in the same initial state. But what normally happens is the cortical activity jumps out here to the right, bends around this turn, does this squiggly line, which is this technical term, and then that's where the animal produces the behavior. Okay? That's what happens normally. Now, what happens when we silence motor thalamus? Does cortex just go silent? It just can't do anything? Or does it generate a bad pattern of activity? So that's what I'm going to show you here. You can see a little movie of this over here as well. It's quite interesting. Motor cortex certainly doesn't go silent. Matter of fact, motor cortex kind of does the right thing initially. It jumps out here to the right in the state space plots, but then it gets out to this particular quadrant of the state space plot where things start to fall apart for the neural dynamics, and it dives down in sort of this neural abyss down here where it will persist for basically as long as the perturbation is on. And this is not a period of inactivity. Actually, the neurons are actually more active in this configuration than they are in the actually control configuration, but it's sort of a neural knot. It's just not doing anything. And then interestingly, we can talk about this later, when you turn off the perturbation, you jump back out to the right, do the squiggly line, and actually the animal will reach for the food pellet. Okay, so this really suggests that this motor thalamus is necessary to generate the correct pattern of activity. Without it, motor cortex does something very different. And you could potentially think about what's happening in here, about what is autonomous to motor cortex. Okay? So it's necessary.
I have a question. Could I ask a question? Sorry, this is from the link. I was wondering if, in case you looked into what the PCs are actually encoding in the two conditions, have you had a look at what because I imagine that in both of these cases, the PCs, what they're encoding must be different, right? Like what is PC1 in blue is not PC1 in green. So maybe the first question is, how do we compare between the two? And second is, do you know what they're actually encoding?
Yeah, so you can do the principal components anyway. This particular one was we did the weights in the control, and then we plotted in the laser trials into it. So the PC1 and PC2 are the same in this configuration. And you can actually do it the other way as well. You can actually do your PCA on both trial types, but this one is done actually on control. Now, what they encode is a little bit difficult, especially when we only have, one trajectory. Like, it's hard for us to really pull this out. Like in the monkey stuff, they can, because they have many behaviors and much variability, they can really ask interesting decoding questions. We're a bit more limited there. We know that PCs 1 and 2 can decode aspects of the movement, such as the velocity of the movement and things like that. But I don't know what each one does. PC1 in the monkey and probably in the mouse is likely a condition invariant component, largely locked to the triggering of the movement, like the big sort of push in order to produce the movement in general. That's what it's been in the monkey, and I think it's probably similar in the mouse. But going beyond that, I'm unable to provide probably a satisfactory answer. Right now we are, yeah, go ahead.
No, I just said thank you for the answer.
Oh, okay. We are doing the current version of this behavior on the animals are now doing 2 different reach targets and 2 different control policies, all in the same animal. And so we could potentially start to ask and untangle what the neural activity does, or the different dimensions and behaviors like that. I just think we're a little underpowered here. Okay, so it's necessary to generate these correct patterns of activity, but actually, is it sufficient? Can you actually use thalamic information to steer around cortical activity? For that, we did sort of the analogous experiment. Instead of silencing motor cortex, what we're actually going to do is stimulate, sorry, not motor cortex, motor thalamus. So we're going to put channelrhodopsin in motor thalamus, and then we're going to put blue light onto motor cortex. And we're going to do this at different frequencies to sort of get like a dose-response curve to thalamic perturbations to motor cortex. And what I'm showing you here, again, on the left, is the neural activity averaged over the trials, and on the right, what's happening in the hand. And these are the different frequencies. So the white line is what's happening in control. The pink magenta line is what's happening at 4 hertz. And then what's happening at 10 hertz is this purpley line. And then the cyan line is what's happening at 40. And so what we really see here in the neural space is a sort of dose-response effect that we get different effects of the neural trajectories given these different thalamic inputs. So we seem to be able to steer around the cortical activity using motor thalamus. And then actually, it's not just junk neural activity that's impotent for driving movements. You can see really similar effects on the arm over here in this graded fashion. So you are able to sort of steer motor cortex with thalamus. Is that what's happening in real life, though? One hypothesis that you might make, that if thalamus is really giving this pattern of activity to motor cortex, is that there should be rich information in motor thalamus about the activity patterns in motor cortex. So in order to answer that question, we try to do simultaneous recordings from both motor thalamus and motor cortex. Now, this ends up being a really tough experiment because you need to find the right parts of motor thalamus. We were able to do this by leveraging these neuropixel probes, just give us a lot of good chances to find the right units, and actually using optogenetics to antidromically identify the neurons along this path, okay? So what we did was we found the units that corresponded to motor thalamus that projected motor cortex, and then we looked at the activity profiles at the population level of thalamus and cortex. And what you can see here at the qualitative level is an amazing similarity between what's happening at the population level in thalamus and the population in cortex. And I don't have time to tell you about this today, but we can actually show quantitatively that there's a lot of information here. For example, I can use thalamus activity not to predict the behavior, but to predict the future state of cortex. And actually, often I can actually predict the future state of cortex from thalamus better than I can do that from cortex, which really suggests there's rich information in thalamus. And if cortex was an autonomous dynamical system, it should certainly be true that I should be able to predict its future state better from itself, which is not always the case. All right, so at this point, what we had sort of concluded was that unlike the sort of role of the spinal cord for things like locomotion, motor cortex does not seem to be an autonomous pattern generator capable of doing all of the interesting dynamics that are required for skilled movements, but it seems to be critically relying on inputs from these other areas from motor thalamus. And remember, motor thalamus is actually made up of many different parts, receiving inputs from many other parts of the brain. And a lot of the work that we're doing in the lab right now is dissecting out these specific roles of all these thalamic channels and what each one contribute to the pattern generation that we see evident in motor cortex. And usually I give this talk and talk about that, but today I thought I'd talk about another side of this story, which is equally as complicated, which is on the output side. So we would really like to know, for these really important dynamics that are sitting in motor cortex, how do they actually contribute to movement? Like how do these dynamics actually produce anything that resembles a skilled movement? This is complicated by the fact that motor cortex goes to actually many places. It goes to basal ganglia, it goes back to thalamus, it goes to the brainstem, and famously, of course, it goes to the spinal cord, which is an old love of mine. And so we would like to understand how these things are receiving this information, processing it, and then using it for movement. This is further complicated by the fact that not only does motor cortex go to all these areas, individual neurons within motor cortex go to all these areas. These are reconstruction of a single neuron from our mouse light project at Genelia. You can see this neuron's axons going to many different targets. And right now, as a field, we don't have a really good toolbox for dissecting out the collateral paths from individual systems like this. Now, there's one target of the cortex where we can attack and try to understand, and that's because of a sort of anatomical gift we were given from this area called the pontine nucleus or the pontine gray. It provides a disynaptic relay between motor cortex and the cerebellum. So it receives most of its information from the cortex, including motor cortex, and then it only delivers its information to the cerebellum. And so, therefore, by understanding this, we might be able to understand what motor cortex is telling the cerebellum and why. Okay? So the first thing we wanted to do was know what kind of information was sitting in the pontine nuclei. There have been very few recordings from this nucleus, especially during movement. That's because it's sort of on the wrong side of the brain here, 10 millimeters down. But we thought that we could get there by leveraging, again, these neuropixel probes and by using optogenetics to help identify the pons. We know that the pontine nuclei is in the densest termination of cortical output, and therefore if we put channelopsin in the output of motor cortex and pinged it off, and then looked for the heaviest recipient field for that, then we'd have pretty good evidence that we were in the pons, which we then later confirmed with histology. Okay, so we can find these right areas, then we can look at what these neurons do. How much time do I have left? Sorry. Okay, I'm going to go quick. So basically, I'll skip this part, but we see a diversity of neurons, but here's a big collection of these neurons. And you can see that a lot of the neurons in pons actually have very transient activation patterns, and they're highly related to the cue, which, we see those in motor cortex, but we also see tons of activity that's tiling over the whole behavior. And you can get further sort of evidence for this by using a GLM to try to understand the firing properties of these neurons. And if you make a filter to either the cue or to the reach, while you can find neurons that have some heavy weights from the reach filter, actually the heaviest weights are from the cue. So this is starting to suggest that actually the pontine gray has an unusual biased information about initiation and preparation of the movements. Okay, so that's starting to be a little interesting. We now wanted to see what the fate of this information is as it sort of goes through the cerebellum. And so we thought one way to do that was actually take control of pontine firing patterns, put in set frequencies of information, and then monitor how they got transformed throughout the entire loop. So the way we got selective control is we made a transgenic line that expresses Cre recombinase only in the pons. Then we put channelopsin selectively in those neurons. We've done this for a variety of different frequencies, but today I'm just going to tell you about one stimulation frequency we put in, which was 40 hertz for 2 seconds. So we're just going to go sort of take a tour through the cerebellum of what happens when we put this 40 hertz of pontocerebellar information in. The first sort of stop at the granule cell layer. These are extracellular field recordings from the granule cell layer. The pons projects directly to the granule cells as mossy fibers. These are these really simple neurons that receive these mossy fiber inputs. And actually what you see in the granule cell layer is a nice entrainment at the 40 hertz that we're putting in, which is expected. We think that the mossy fiber granule synapse has high fidelity, and this is what we expect. So let's move on now from the granule cell to the Purkinje cells, which is the next station. Granule cells provide these parallel fiber excitatory inputs to the Purkinje cell. And if we look at the responses to this 40 hertz stimulation, we see lots of neurons that are driven at 40 hertz, seem to be nicely entrained. But we're starting to get some neurons which show very different response properties. For example, we start to see these pauses in the activity patterns at the onset of this 40 hertz stimulation. Okay, so moving quickly to the next station, the Purkinje cells project an inhibitory projection to the deep cerebellar nuclei. Here you start to see even more of a transformation. You start to see a lot of transient activations, which makes sense since this is an inhibitory projection, but much less tonic neurons here. You do get some neurons that are entrained, but it's less in the population. And let's do this to the final station, which is all the way back to the cortex. And here we got a kind of a big surprise. On the right here is what I'm showing you is what happens in the cortex. Now almost all the inhibitory responses are gone. Almost all the tonic activity is gone. There's no really entrainment. It's mostly just this transient activation. And that's quite different than what we see even in the output of the cerebellum, the deep cerebellar nuclei. Okay, so this is really interesting. We put in 40 hertz of 2 seconds of information, and what comes out by the time it gets back to the cortex is a transient activation period. So we're really interested in who are these neurons that are receiving this transient activation. So, and who for us means what do they do in the behavior? And if you compare those transiently activated neurons to their neighbors who are not transiently activated and look at what they do during the behavior, they're actually firing much earlier in the behavior relative to these neighboring neurons. And this is starting to build up this argument that maybe the pons, which is carrying all this initiation signal, no matter what I put through the cerebellum, I get like a transient activation, and maybe that's helping kick off cortex. Okay, so is that what it's doing? We want to now know what the pontocerebellar pathway is doing for the movement, so let's perturb it. We've done it many different ways, but I'm going to show you this perturbation, 40 hertz for 2 seconds, because we can leverage that in a minute. So when you do that, actually you get a really nice effect on behavior. And as you can see, what we normally see is dysmetria, which is a mistargeting of the food pellet position. Here you can see attempt over attempt while the laser is on. They're just overreaching in this particular case, quite wildly, actually. And then as soon as we turn this laser off on this particular trial, they're going to immediately recorrect and target the food pellet. So it seems like it's having this nice effect on the targeting of the food pellet. You can see this right here. Without ever going back to the perch, they just now can correctly target the food pellet. And you can see this sort of dysmetria if you look at the success rates of these animals and of the standard deviation of the hand position at the grab position. And you can also see other kinematic features affected. For example, one thing that's relatively consistent is an effect on speed. Okay? So we really wanted to understand really why this was happening. And so we went back to our neural recordings and we said, okay, what's going on at the neural level, both in the cerebellum and in the cortex while the animals are receiving this laser and then mistargeting the food pellet position. And as you can see here, neural activity actually looks pretty normal. Like it's remarkably similar. This is the cerebellar nuclei and this is the cortex. I mean, they are shockingly similar. And remember, these are the same neurons I just showed you that can be robustly affected by the laser alone. So this really suggests to us that when the animals are producing behavior, they can quickly sort of like digest away all these sort of effects of the pontocerebellar pathway, and the behavior and the dynamics underlying the behavior is quite robust, even to that very powerful perturbation. And we would love to understand the mechanisms of that robustness. Now, there are some differences between these neural activity patterns, and I'll do this rather quickly. If you ask if what is different between the neural activity patterns, explain the behavioral differences, you can. So this is a decoder, either based on the DCN or on motor cortex, and what's happening to the hand, and you can observe these reductions in speed from either. So most of the dynamics are robust, but what is not robust to this laser perturbation seems to be really important for setting fine features like the endpoint accuracy and the velocity profiles. Okay, and I promise I'm almost done. I put you in a really weird place though. I just told you like I've got all these signals, like initiation signals, and I just showed you a bunch of execution deficits. So like why did I tell you this? And we were really surprised by this too. And it's not true that we don't see initiation deficits. Some animals actually during the PN stimulation cannot initiate, And you can see a couple of those animals over here. If you do a pharmacological inactivation of the pons, you can actually affect the reaction time. And actually, some animals will weirdly actually will trigger the whole behavior off of the laser alone. So there is some evidence that this pontocerebellar pathway could be involved in initiation, but why weren't we seeing it more robustly? And we thought about this for a little bit, and we thought maybe if this really is a trigger signal, maybe it's actually very easy to adapt away from this perturbation because it can be very simple. And what I'm showing you here is like animals that have seen these perturbations for many, many trials. So we decided to go back, retrain animals, never show them the laser, and give them a block of trials, the first block they've ever seen. And there you see a big, big consequence. These pink trials down here are the first trials the animal's ever seen with this perturbation, and they cannot initiate the behavior. But then, 15 to 20 trials later, they pop right back to normal behavior. So we were really shocked by this. This is not the normal sort of timing of motor learning in cerebellum, which usually takes hundreds and thousands of trials. This is happening over 15 trials where you go from absolutely no behavior back to behavior. So we wondered how this happened. We looked at the neural activity again, both in the cerebellum and the cortex. What I'm showing you here is the cerebellum. This is the first three PCs that explain most of the variance. You can see these nice patterns of activity over these trials. And then what I'm showing you down here is the first 15 trials where they see the perturbation from the pons. And you can see the neuroactivity is not normal here. Something really egregious is happening. But also you can start to see, this is even truer at the single neuron level, which I don't have time to tell you about, some reshaping of this neural activity. And then eventually it pops back, and then the animal's doing the behavior again. So the cerebellum seems to be able to quickly adapt to this wild perturbation that we're putting in, 40 hertz over like the entire population of Ponti neurons, and get back to the normal dynamics underlying movement, basically. Now, what is the cortex doing over these 15 trials? This was a big shock to us. They're basically silent over these 15 trials. So somehow, while the cerebellum is working out this horrendous insult that it's dealing with, it's actually dissociated itself from communicating from the cortex. And I think this is a really interesting potential mechanism that when you're doing this adaptation, maybe you actually don't want to fully communicate along this loop so that you can preserve all the hard-won plasticity happening in the cortex. But the mechanism of this dissociation is not clear to us. So this is, I think a real interesting evidence of resilience in these circuits. We would love to understand this resilience because it is not clear to us how you get back to a good behavior, a good neural behavior, and good actual behavior so quickly. And what are the learning and teaching mechanisms for this? How does it know which direction to go? And actually, for this question and many other types of perturbations in the brain, we're becoming sort of obsessed with that notion. So I'm going to end there. What my lab is trying to do is try to understand skilled movements. We're trying to understand how all these parts interact in order to generate these pattern generators that are so critical. And then I have to thank the people who did this work. Jay and Jeremy, who have come down with me to UNC, did almost all the experiments I showed you today. And Matteo and Britton, who have both moved on, I can't find Britton's name in here. There it is. They did all of the awesome analysis. So thank you.
2 more minutes, so please feel free to ask questions over there below.
Really nice talk, Adam. You showed a really interesting, what you call dose-response curve with the motor cortex with different frequencies. Did you try that in the pons, and did you see things at the lower frequencies that seem to be more effective in the motor cortex?
Yeah, excellent question. I'll repeat it just in case someone didn't hear it. Did we replicate a dose response, or did we retry a dose response curve in the pontocerebellar manipulations? Not as carefully. We have tried it. It's something interesting about it. Sometimes lower frequencies actually perturb the system worse than the higher frequencies in that system. And we haven't done enough of it. It's actually on board for future work. But there is something there. And so it would be really interesting to see, to monitor this pathway, both at the sort of short time scale and this more adaptation time scales for different frequencies. Because there, as you probably thinking in the background, there's really interesting literature about coupling oscillations between cortex and cerebellum, and are there sort of sweet spots for that? And this is a really tough experiment, and I have a hard time convincing Jay or Jeremy to do these experiments ever again. That is really it's on our sheet for things to do. Yeah, great question.
Hi, we have a question from Lena Ting in the virtual audience.
Yep.
Hey, Adam, that was awesome. I had a thought about these frequencies also because, in the lower limb CPG, you can stop it by blocking the hip stretch information. So you can basically change the transitions from one state to another by manipulating the sensory feedback you get from the angle, since there's different phases. And so I'm wondering if the 4 hertz was effective in the arm because it's on the order of the frequency of the movement. And so there's something about having the time transient input to kind of keep that rotational trajectory going to complete the reach. And my second thought, is that in the PN stimulation, you have this tonic activity, and it looks very much like cerebellar adaptation, where like maybe you stimulate, you have a huge loud noise and you can't hear it, and then you basically learn to filter it out, and you can hear it again pretty soon. Those are my thoughts about the 2 types of frequencies.
Yeah, so I kind of went over this a little bit quickly, but the 4 hertz in thalamocortical transmission, it will cause a bubble in the movement. So it basically he'll hang and then go and continue on. He doesn't forget to do the action, but it will affect. It causes a bubble in the neural activity in motor cortex, and it causes a bubble in the trajectory in the arm as well. So it's just that 10 and 40 puts you in such a bad state that you are locked into a different position and are unable to fight through it. But if you think about 4 hertz over the length of one of these movements, which is like 300 milliseconds, like you're basically only getting 1 stim in the movement. So 4 hertz is still pretty effective. It's just that 10 and 40 like pull you to a part of the state space that you cannot break through from. So I think actually all the frequencies are potent for the thalamocortical stimulation. Now, the pontine stimulation, whether they, whether they interpret that as sort of like as just a giant increase in noise, I need to think about that. I don't have like a coherent thought about what that really, like how to distinguish that between alternative hypotheses there. It is clear that, one interesting thing is actually most of the action is in PC3 there, and like what PC3 is like actually related to in the movement would be interesting to maybe explore to try to figure out exactly what is going on there. I don't know. I don't have a good answer for that second question.
Hey, Adam?
Yeah.
Hey, kind of popping up a level, here it's very striking that the dynamics of the movement are almost perfectly matched by the dynamics of the brain. And do you think, as the movements get more and more complex, that there might be more of a dissociation, where there's maybe a limit on the complexity of the dynamics that you can generate in motor cortex, or vice versa? Yeah. Right? And maybe that might also, tease apart some differences between thalamus and motor cortex and some of these other brain areas.
Yeah. Yep. So, I think at the nature of your question is if we drive up the complexity of the behavior, could we get activity that actually is not so well, is basically abstracted from the movement? And I think that's possibly true. I think you'll still have dimensions in motor cortex that will nicely represent the movement. There's almost no way around it. Even if you're not even driving the movement, you'll reflect the sensory inputs. But you might actually gain some specific dimensions that in the neural activity that are quite different and quite abstracted. That's certainly the case. So that's one of the reasons why we have these animals do these 2 different control policies. Are there like sort of dimensions that are just about which control policy you're in, which is kind of a version of that. We'll probably know in the next few weeks. But I think that's possible. I think if you look at the monkey data, maybe in a monkey, those sort of more conceptual dimensions might be in other areas, not in motor cortex. But in the mouse, I think it's fair game because like everything might be squished into this one region.
Hi, great talk. Thanks so much. I was wondering with the thalamic inactivation experiment, whether you think that any kind of disruption to thalamic activity statistics would have driven an impairment in the behavior, or that it's specific to inactivation.
Well, the 41040 is like another version of that. That's a stimulation, and that was clearly effective. We've done lots of like unpublished experiments where like we inactivate for like just little short bursts, and actually you could like slow the arm down. Like you can. It's like perfectly like yoked to motor cortex. So almost everything you see in thalamus and every perturbation you do, you can get interesting and disruptive effect in motor cortex. Yeah, I mean they are well coupled there.
I have a question. You seem to have sort of taken premotor cortex out of the equation where a lot of people think planning takes place, and to some degree there's still an ongoing interaction during execution. So going back to your piano player that you showed, I mean, they have to solve problems at different time scales and complexities, right?
Yep.
Playing each note with the fingers, but also planning how intense they're going to play it. And then they have things about melodies and rhythm and really high-level concepts. So where do you think motor cortex comes into that, and where does, say, premotor cortex come into that?
Yeah, I mean, I can't find premotor cortex for the arm and mouse. I mean, like, in the experiment I was talking about before, where we had them doing different control policies and different choices, we have inactivated everywhere outside of motor cortex, and none of those areas seem to affect the choice probabilities or like any of those sort of higher level aspects of the decision. So one possibility is that they, like in the mouse, like they all sort of got wrapped into motor cortex, or more importantly, and probably more probably, in these loops, including the basal ganglia and the cerebellum, that they're all sort of wrapped, but then they basically are all sort of centered on motor cortex. But I think in the mouse, right now we have and we've tried hard to like look at prefrontal areas and parietal areas for any other thing that can affect even fine features of the movements. And for cortical areas, we can't find anything.
That's the RFA and the CFA, so you don't see those rostral and caudal forelimb areas?
Well, we see them. I mean, we are likely affecting both in almost all of these experiments. And so when we have tried to, so light actually spreads considerably when we do these experiments. So it's hard to limit it in the experiments like with a transgenic animal. We have gone back and tried to do RFA versus CFA experiments for the reach task. And actually, you don't really block movement there. And I think it's because while there are some separations between RFA and CFA, they are highly similar networks and actually maybe redundant with one another. And so if you take one out, the other one can very quickly overtake the whole function. So we actually don't get much phenotype if we restrict to one area or the other, and we've done it in both orders. So I don't know. And there are stories out there in the literature that RFA and CFA are potentially doing different things, where the CFA is doing the reach and the RFA is doing the manipulation of the food pellet. We haven't seen any evidence for that. We have seen evidence of like sensory cortex being really critical in evaluating whether the animals actually got the food pellet in their hand, but that's a quite different thing.
No further questions? Then let's thank Adam again for a really inspiring talk.
Thanks.